Pleasant morning, everyone. We're here today to share with you some of our found published research that contains 5 locals and 5 international research. Before anything else, let me introduce myself first. My name is Crystal Ayagbalog from Chem 45B Group 1. So, research is a creative and a systematic work undertaken to increase the stack of knowledge. It also involves the collection, organization, and analysis of information to increase the understanding of a topic or issue. Research develops understanding and decision making. It is the most valuable tool to understand the complexities of a problem, disprove lies, uphold truth, and build on the create of knowledge that is reliable and authentic. Conducting research develops a better understanding and enhanced decision-making capabilities. So my co-members would like to share with their 5 locals and their 5 international research that they found on the internet. So stand up straight, keep your hands out of your pocket. Now I'd like to look at our next topic that might help you to get some knowledge and some ideas, so keep on watching.
Good day, my name is Apple Emboliasis, and today I am going to discuss with you or present to you our 5 international published research that we have gathered. So the first study is the roles of lipids and nucleic acids in HIV-1 assembly. The authors was Aina Al-Fadli and Eric Berkles. The study was published on May 2014. It was conducted in Department of Molecular Microbiology and Immunology, Oregon Health, PS Sciences University in Portland, USA. The instrument used was experiment. For the analysis of the strong and weakness parts of the research, the strong part of the research, the researcher reviews the functions of retroviral MA proteins with an emphasis on the nucleic acid binding capability of the HIV-1 NA protein and its effects on membrane binding. In the review, it focuses on the role of MA binding to RNA and summarizes the importance of GA-GMA interactions with RNA for HIV and other retrovirus, with the hope that this comparative approach can shed more light on our understanding of the importance of these functions and ways of inhibiting that role. For the weak part of this research, the authors declared that the research was conducted in the absence of any commercial or financial relationships that could be construed as a potential conflict of interest. The second study is the chemometric identification and nutritional evaluation of 3 species of Luginidae from the Amazonian Atlantic coast based on fatty acid profiles. The authors was Edeline Santos de Amilda and Evaldo Martins de Silva. The study was published on October to December 2016, and the study was conducted in Amazonian Atlantic coast. The instrument used was the test or experiment. Now, the analysis of the strong part and the weak parts of this research. For the strong part, the researchers' strong part of the study was to determine the proximate compositions and FA profiles of heart, brain, and muscle tissues from 3 fish, which are the Lujanus porphyrius, Lujanus tenebris, and Cyrus cineros, that are captured from the Amazonian Atlantic coast. They show different available data that can be used in analyzing fatty acid of each different species. They also use a chemometric tool in performing nutritional analysis of each fatty acid profile in order to discriminate among the species, and it successfully showed the results that the Lujanus tenebris had the highest protein content, while the O. cyrinorus had the highest lipid content of each fatty acid profile. Due to its good sources of protein and PUFS, it showed interest to consumer for human consumption, which result of this study confirmed the importance of the chemometric method for the identification of species and fish stocks. Now for the weak part of this research, the researchers use many abbreviations and the other unfamiliar symbols and words which make some of the statements to be confusing. They also use small fonts and sizes. They also didn't apply any paragraph spacing that makes the statements become unreadable. The third study, lipid contents and fatty acid composition of some New Zealand freshwater finfish and marine finfish, shellfish, and grouse. The authors was Peter Blade and Dennis R. Body. It was published on March 2010. They conducted it in New Zealand Ministry of Agriculture and Fisheries. The instrument used was sampling collection. Analysis of the strong and weak parts of the research. For the strong part, the recent studies of the nutritional value of the edible parts of fish in the New Zealand fishing areas include lipid and fatty acid composition of shellfish, was reported on the nutritional composition of snapper, mussels, scallop, and barracuda, blue mackerel, monkfish, blue, and silver warehouse, respectively. Blay reported on lipid and fatty acid contents of blue mackerel, jack mackerel, and red cod. Body studied cardinal fish, javelin fish, look-down dory, retail, ribalda, and sea perch. But we wish to extend this data to include fish species not previously recorded, as well as report data on previously analyzed species caught in different areas at different periods. In the present work, that the fatty acid composition of the edible parts of 6 water finfish, 60 marine finfish, 9 marine mollusks, and 3 crustaceans are described. In addition, 6 raw oils have been investigated to provide nutritive data per use by nutritionists and consumers. The weak part of this research, marine wax esters were detected in the fillet of orange roughly, Oreodori, and smooth Oreodori. And in all finfish roe study, the commonly found distribution of the major fatty acids, moieties, and marine oils was observed in all species examined except orange roughly. The polyunsaturated fatty acid content of each species is given. For the fourth study, into the deep, new data on the lipid and fatty acid profile of redfish, sea bass, Manila, inhabiting different depths in the Imanger Sea. The authors was Victor P. Varonin, Nina N. Nirvana, Tatiana Rogobin, Demetril V. Artemendol, Alexei Alexel M. Oryuv, and Sventlan A. Mazina. The study was published on May 2021. It was conducted in Russian Federal Research Institute of Fisheries and Oceanography. The instrument that they used on this study was sample collection and schematic map. For the analysis in the strong and weakness parts of this research, the strong parts was the lipids are the principal structural and energetic components of marine organisms. The primary depots of which are muscle tissue and the liver. Studies have revealed a relatively low content of TLS in the muscles of big redfish, males and females, which dry weight respectively, which is regarded as a biotechnological characteristic of redfish. In terms of the fat or protein ratio, the North Atlantic Disebes can be classified as a semi-fat fish, or the fat content is 3 to 6%, while fish such as the Greenland halibut and the Atlantic salmon are described as fat. Fatty acids as integral components of lipids are highly polyfunctional. Their quantities and composition are directly dependent on the organism's physiological status and the combination of environmental factors. The high content of essential omega-3 polyunsaturated DHA and EPA we observed in big redfish mussels comprises the species characteristic trait, and as demonstrated previously for redfish and other deepwater long-living fish species, the quantities of this acid increase with age. For the weak part, a study of the lipid and fatty acid profile in the mussel peak redfish males and females from different depths in the Erminger Sea showed that there was no significant change of the total lipid content with depth. The total lipids dynamics with depth trended in the opposite directions by decreasing in males and increasing in females, indicating sex-specific differences for the lipid level compensatory responses to vertical movements in the water column. Vertical migration, this was corroborated by the prevalence of cholesterol esters over triglycerides, and the high contents of wax esters. Lastly, lipids in marine ecosystems. The author was Christopher C. Parrish. This study was published on April 2013. It was conducted in Department of Ocean Sciences, Memorial University of Newfoundland, St. John's, NL, Canada. The instrument they used was sampling collection. Now, for the analysis in the strong part and weakness part of this research, the strong part was lipid analysis, lipid nutrition, lipid oxidation, membrane fluidity, molecular biomarkers, multivariate data analysis, and quantification of trophic relationships. These authors used primer in multivariate ecological research to investigate algae feeding, amphipods, and sea urchins. This is a software package for analyzing ecological data marketed by Primer Enterprises. They use analysis of similarities, a non-parametric approximate analog of univariate, which ANOVA tests, to provide a measure of similarity between groups and similarity of percentages analysis, or SIMPER, to examine the contribution of each fatty acid to average resemblances between sample groups. SIMPER identifies that which is primarily providing the discrimination between observed sample clusters. For the weak part, the principal components analysis has a strong following as it permits data reduction and objective interpretation of results, but several more sophisticated multivariate analysis which are more quantitative are emerging too, therefore vague. That's all. Thank you.
Thank you, Ms. Valiasas. So I will give you the 5 local research that can be found in the internet. To begin with, species composition, abundance, and catch trends of brown scads Decapterus spp in Iligan Bay, Northern Mindanao, Philippines by Cesaria R. Jimenez, Lionel L. Molina, Jerry P. Garcia, Marife B. Quiñones, Hariken T. De La Rosa, Jeanette J. Samson, and Margarita C. Paghasian. And it was published on May 2020 at Mindanao State University at Naawan, Misamis Oriental, Philippines. And the instruments used is records, and the DOI number is on the screen. So, the strong part of the research. The researcher provides information which is essential in determining management options to improve their production in the future and provide basic information on round scads resources in Iligan Bay that forms part of scientific basis in crafting a suitable fisheries management plan. The paper presents the composition, abundance, and catch trends of brown scads in Iligan Bay. The generated information is an important input in a site-specific ecosystem-based fisheries management plan for round scads in Iligan Bay. So the weak part of the research, as I noticed, the results of this 1-year monitoring on the catch landings only provided initial insights on trends but are already useful information that can be used as framework for site-specific ecosystem-based fisheries management plan for round scads in the bay. However, further dockside monitoring of longer duration is recommended to generate a more comprehensive picture of the status of the resource in Iligan Bay. The second research is growth in fatty acids, profiles of Halophothora vescula and Salispina spinosa from Philippine mangrove leaves by NALE Cagimbal, MKP Devanadera, RM Binet, KHV Arafiles, K Watanabe, P Ache, GR Didiles, and it was published on July 2019 at Philippine Mangrove Forest in Aklan, Quezon, Davao del Norte. And the instruments used is sampling collection, and the DOI number is on the screen. So the strong part of this research is studies on marine source fatty acids have gathered significant interest recently as an important component of aquaculture, feeds, and of biofuel production. Of the organisms capable of producing fatty acids, marine oomycetes are a promising model organism. One group of marine oomycetes are the Halophyta spp, which is known to have an important role in leaf decomposition, thereby changing the plant debris into exudates, which are usable to consumers in the mangrove ecosystem. Results relieve the presence of different fatty acids such as arachidonic acid, linoleic acid, and vasenic acid when grown on V8S and PYGS media. This study is the first observation of fatty acid from S. spinosa and H. vesicula from the Philippines. And the weak part of the research is due to the increasing demand for an alternative source of fatty acids, species of oomycetes isolated from select mangrove forests in Luzon, Visayas, and Mindanao were analyzed for their fatty acid contents. Prospects for industrial important fatty acids make these oomycetes all important to study in applied microbiology in the Philippine setting where those structural simple microorganisms abound. The third research is the fatty acid composition of 5 candidate aquaculture species in central Philippines by Ogata Hiroshi Y, Imata Arnel C. Garibay, and Esteban S. Furiota Hirofumi, and it was published on July 2011 at Central Philippine University, Iloilo City. And the instruments used is observation, and the DOI number is on the screen. The strong part of the research is the researchers were able to provide credible and presentable data which they use the different types of dialysis which ARA, EPA, and DHA are present in determining the fatty acid composition of 5 candidate aquaculture species that results the total lipids and hatchery produced eggs and newly hatched larvae of mangrove. Red snapper unexpectedly had equal levels of ARA and EPA. It also showed that in the central Philippines, the 5 species appears to have intermediate or high ARA and DHA level with low EPA level, but in the present result, it shows that ARA is not a minor component in the tropical species and suggested that ARA is much more nutritionally important for egg development and larvae growth in tropical species than in cold water species. The weak part of this research is that the researchers provide different statements about the composition of fatty acids in 5 species, which results in confusion and could take much time in understanding the study. It has many observations which cannot be easily identified with the meaning of the words. The researchers used unfamiliar words and use small size text that makes each statement unreadable and confusing. So the next research is about fatty acids in 6 small pelagic fish species and their crustacean prey from the Mindanao State Sea, southern Philippines, by Anthimir Bicoy, Metelio, and Ana Arlene Aspiras Ea. And it was published on August 2014 at Mindanao Sea, southern Philippines. And the instruments used is sampling, and the DOI number is on the screen. So the strong part of this research is in the strength of this study that they were able to provide supporting incredible data on the study. They use sampling as a method of research, and it gives information on the study of fatty acids in 6 small pelagic fish species and their crustacean prey from the Mindanao Sea in the southern Philippines. And the weak part of the study is that the weak point of this research is that it does not include the local names of 6 small pelagic fish that should be easy to determine for every local fisherman in the Philippines. Also, they lack information citing the area where they conducted the research. It would be helpful if they include the specific location that should help to be easy to visualize from the reader. And the last research is survival and settlement rates of Haliotis asinina larvae at different salinity level by Jean Rose Hilario Makirang and Romeo Caturao and Jesse H. Makirang and Fiona L. Pedroso, and it was published on July 2014 at Iloilo, Philippines. And the instruments used in the research is experimental, and the DOI number is on the screen. The strong part of the research is that the study determined the survival and settlement rates of donkey ears abalone or the H. asinina at different salinity levels. It also determined the optimum range of salinity for better larval survival and settlement for donkey's ear abalone or the H. asinina. And the weak part of this research is that assess how long H. asinina larvae can survive and settle after a long exposure to low salinity until the juvenile stage with the appearance of first respiratory pore, and to evaluate larval shell growth of H. asinina larvae reared at 24, 28, 32, 36, and 40 TPT. And that's all the local research that we found on the internet.
So, those are the published local and international research that we found on the internet. So, for the last part of this video, we would like to share with you our individual insights and thoughts about our future plans on our research. So, for my future research and for my future study, I am focusing on innovation and contribution of lipids in food and in fish nutrition. Lipids contribute to many desirable qualities to foods, including the attributes of texture, structures, flavor, and color. Generally, the quality of food is closely related to the quality of their lipids. Very often, the degradability and the alteration of lipids are the main causes of the loss of quality of food. Lipids are important in energy storage molecules in most organisms. However, equally or even more important, specific lipids play critical roles in a wide range of physiological processes. Lipids is important for our marine species, especially to the fishes.
Good day, everyone. My future research plan focuses on using modernized methods and strategies in sustaining and improving fisheries and aquaculture production to benefit the poorer household in the Philippines. In this study, it focuses on how the poorer household in the Philippines will be benefited in improving the fisheries and aquaculture production using modernized methods. It gives me an interest because using modernized methods will be much easier to do the job, and it will also be easier to manage and monitor the production of fish. If this will be conducted, lots of people, especially those poorer households in the Philippines, will greatly benefit in regards to job opportunities and in sustaining their needs.
For my future research or for my future study, I am focusing on the nutritional value of lipids and fatty acid of the edible parts of fish. Lipids and fatty acids are essential for all life on Earth. They play many important roles in maintaining the health of an organism. Arguably, the most important function lipids perform is as the building blocks of cellular membranes. Other functions include energy storage, installations, cellular communication, and protection. It has a huge purpose in the aquaculture because it is the major organic constituents of fish, and they play major roles of sources of metabolic energy for growth, including reproduction and movement, including migration.
Hello, my name is Apple Emboliases, and today I am going to share with you about my future plan with my research. I'm really quite interested to study the echinoderms, specifically the diversity of echinoderms, and because they are very capable to regenerate their muscles, their tissues, like that. And I want to know that deeply more. And since there are some of the echinoderm species that we don't know much, because they are not very exposed in the public, like in the market, like that. They are not like the fishes that we can see in the market. And yeah, I wanted to know that. And also, if I am going to conduct this study in the future, I wanted to stick with the comparison of the echinoderms that they used to live before and in the present, like that. And that's my goal in my future research. That's all.
Hi, I am Curtly Asuncion, and my future research plan is fisheries habitat investigations. This research is all about marine species harvested by the commercial and recreational fisheries require habitat to grow and reproduce. The fishing community and other stakeholders identified several habitats of interest, including coastal lagoons and estuaries around Cape Cod. And this research is supported by the Friends of Pleasant Bay and has set up stage for long-term monitoring to detect changes in the system, as well as focus studies on specific ecosystem indicators, such as the presence of tropical fish via a collaboration with the Gulf Stream Orphan Project. And this project was part of a larger interdisciplinary, multi-scale ecosystem assessment of Pleasant Bay.
Hi, I am Edwardson de Barquero. As a fisheries student, I would like to focus my future research on restoring damaged coral reefs using mass coral larval reseeding. We all know reef corals are critically important because they build the primary reef framework, supply essential habitats for thousands of fish and other species, and provide direct energy and other nutrient inputs to the ecosystem. But the loss of substantial live coral cover on reefs fundamentally through overfishing, eutrophication, and other disturbance arising from human activity has seriously disrupted their ecology. Continued human pressures on reefs results in further decline and ecosystem collapse. But perhaps my future research would help alleviate this situation through mass reseeding with coral larvae.
We really appreciate your attention today. Thank you so much, guys, for your efforts, attention, and interest on listening with us. And we would like to finish this presentation with an inspiring birit for Ma'am Magnagi. Ma'am, we are like DNA structure. You are our adenine and we are your thymine. Thank you and God bless.